Application of plant tissue culture. These are the applications about the plant tissue culture. First one is production of virus-free plants. Second one is somo-clonal variations, production of somo-clonal variations. Third one, secondary metabolites. Fourth one, synthetic seeds. And the next one, crop improvement, micropropagation, genetic transformation, storage of germplasm called cryopreservation, hairy root culture, transgenic plants, and production of haploids. See, these are the applications of plant tissue culture techniques. Production of virus-free plants, production of somo-clonal variations, production of secondary metabolites, production of synthetic seeds, crop improvement, micropropagation, genetic transformation in plants, storage of germplasm called cryopreservation, hairy root culture, transgenic plants, and production of haploids. So first point is production of virus-free plants. See children, during the tissue culture technique, we selected explant from the shoot tip only. The shoot tip cannot infected. The shoot tip cannot infected. They are surviving cells. The surviving cells are selected as a explant. Automatically, we selected healthy cells from the plant as a explant. They are automatically produced virus-free plants. Production of virus-free plants means during the tissue culture technique, we selected virus-free tissues as a explant. In plant pots, virus-free tissues are present on the shoot tip. The shoot tip part cannot infected. The shoot tip parts are cannot infected by the virus cells or any other microorganisms. So automatically the fresh healthy cells are take as a explant. Automatically we can produce virus-free plants by the tissue culture. And the next one is somo-clonal variations. Somo-clonal variations are produced by the protoplast fusion culture. Protoplast fusion culture produced hybrids called somo-clonal variations. So here it is, we take two different types of healthy plants. The taken two different types of healthy plants containing two different varieties of needed characters. The two needed variety characters are present in only one plant. The plants containing two hybrid characters, one parental character, another parental character expressed in the next generation by the production of somo-clonal variations. So we take protoplast from one plant, we take another protoplast from one plant. These two protoplasts are fused to form one hybrid cell called somo-clonal hybrid cell. The somo-clonal hybrid cell cultured on the nutrient medium, it develop into callus. The callus added growth hormones, it develop into organogenesis. Then organogenesis can produce new plantlets. The new plantlets are produced new varieties of characters, new varieties of two plant characters present in only one plant called somo-clonal variations. We produce rapidly somo-clonal variations by the tissue culture technique. And the next one is synthetic seeds. Some plants cannot produce by the seeds. Some plants cannot produce by the produced seeds, flowers. So that plants generations are stopped. So based on the tissue culture technique, we produced synthetic seeds. Vegetative parts of plant pot. Vegetative parts of plant pots are mixed with sodium alginate chemicals. See here, it is sodium alginate chemical. The shoot parts are root parts or leaf parts. The leaf parts or shoot parts or leaf parts are uh, treated with sodium alginate solutions. Automatically they are converted into seed like structure. The seeds are placed in the normal plantation in the normal soil. They can also develop into new plantlet by the synthetic production of seeds. During this tissue culture techniques, callus is developed during the incubation. If we add growth hormones to the callus, they cannot develop into root system and shoot system. At that time, that callus cannot develop into organogenesis, that cannot develop into organogenesis tissues also. Covered with sodium alginate chemicals, they act as a synthetic seeds. So some plants cannot produce seeds. That plants can produced by the synthetic seeds by the sodium alginate chemicals. So these are similar to the normal seed like structures. They can also develop into normal plantlet by the plantation in the proper environment. And the next one is genetic engineering, crop improvement through the genetic engineering. So which we need good characters of genes, that elected genes are transferred into the plants. Elected genes are transferred into the plants by the genetic engineering. Automatically we gain good harvest from the crops, good yield from the crops. So by the genetic engineering means it is a part of tissue culture. It is a part of tissue culture. We can easily produce crop improvement through the genetic engineering. Which gene is needed, which gene is helpful for the crop development, that genes are elected by the special mechanisms by the rDNA technology. That selected desired genes are transferred into the selected plant, host plant. Automatically the selected host plants gain new good characters, gain good characters. What are the good characters here? It is crop improvement. Crop improvement characters are compulsory expressed in the plants called crop improvement through the genetic engineering. And the next one is micropropagation. Micropropagation means within a short period, within the short time, we can produce large number of similar type of plants, large number of similar type of plants by the micropropagation. It compulsory during the tissue culture, we add growth hormones to the callus. The callus differentiated into organogenesis like root and shoot. When we observe the callus under the nutrient medium, the callus is separated by the special mechanism as a cells. That cells are transferred into another nutrient medium. They are also, that cells also develop into nutrient, the cells also develop into callus on the nutrient medium. We can produce a large number of callus. Again, the callus divided into cells. That cells are transferred into the nutrient medium. The nutrient medium containing number of callus. So we can produce large number of quantity of plants or plantlets within a short period, within a less place, called micropropagation. It easily possible by the plant tissue culture techniques. Within a short period means we take only 1 month or 2 months of time. During this time, we take one small length of area. We can choose only one lab room or one seminar hall or on uh, one incubation room. They are kept under the incubation in these rooms. Automatically, we gain large number of lakh of crores of plantlets are produced by the micropropagation. Incubation halls like laminar air flows, incubation chambers, it applicable in the micropropagation. And the next one is genetic engineering plants. Genetic engineering plants means we need desired genes. It possible by the DNA technology or DNA technology. Selected desired cDNA, it transferred into the new host plant. That host plant cultured on the nutrient medium, they can develop into new varieties of characters. So these type of plants are called genetic engineering plants, genetic transferred plants. which gene the gene is desired genes is selected from the other plantlets or other plant cell organelles. The desired genes are transferred into the new host plant. That host plant cell cultured on the nutrient medium, it can develop into new plantlet. The new plantlet containing desired characters. What are the needed characters in the selected desired gene? The selected desired gene containing these characters are expressed in the next generation of the plants. And the germplasm storage. Germplasm storage means in future days we followed genetic storage banks. Genetic storage banks means some endangered endemic species germplasms are stored in a liquid nitrogen at 196 degrees Celsius. At minus 196 degrees Celsius in a liquid nitrogen, any germplasm, any genetic materials stored as a long time, long duration. So automatically when we need that gene, that genes are selected from the genes are isolated from the gene banks. This process is called cryopreservation. Cryopreservation is also called germplasm storage. Endangered endemic species cytoplasms, protoplasms, genes, all types of cells are stored in a minus 196 degrees Celsius of liquid nitrogen solution called cryopreservation or germplasm storage. And the next one, hairy root culture. See children, hairy root culture is nowadays it is used in modern agriculture techniques. Hairy root culture. Hairy root culture, it is the culture product produced after the infection of explant and cultures by the germ, gram negative soil bacteria. Gram negative soil bacteria. The soil bacteria is called Agrobacterium rhizogenesis. Agrobacterium rhizogens produced negative soil gram bacteria. When we added these agrobacterium to the explant, automatically the explants are produced only roots. The naturally produced root, they are mainly healthy in conditions. They can produce secondary metabolism by the root hair culture. Some plants are produced rapidly produced secondary metabolites. The secondary metabolites like chemicals, alcohols, phenols, phenolic acids, like other types of chemicals also produced by the roots. The specialized chemicals are collected from the roots. The roots are developed by the hairy root culture by the adding of gram negative soil bacterium. That gram negative soil bacterium is also called agrobacterium rhizogenesis. Rhizogens, when we add it to the explant means it develop into callus treated with these rhizogens, agrobacterium rhizogens, produced automatically only roots. We cannot add growth hormones. We add only agrobacterium rhizogens chemicals. The rhizogen chemicals are added to the callus. They can develop into only hairy roots only. That hairy roots are produced to secondary metabolites. That secondary metabolites are commercially utilized by the human beings. And the next one, production of haploids. Androgenesis. It is also called androgenesis. When we take pollen culture, the pollen means haploid condition. Pollen grains are haploid conditions. The pollen grains are taken as an explant under the nutrient medium, on the nutrient medium. It develop into only haploid plants. That haploid plants containing only single set of chromosomes. The single set of chromosomes can express all the characters, not suppress genes. All genes are expressed external characters. That external characters are easily identified by the pollen culture or haploid culture called androgenesis. In this androgenesis, which genes are containing good characters, that characters are completely expressed in the generations of the plants. In similar anther culture also, anther cultures means pollen mother cells. The pollen mother cells are cultured on the nutrient medium. The pollen mother cells develop into pollen grains. The pollen grains also develop into plantlets. This process is also called androgenesis. By the pollen culture, by the anther culture, we can produce similar types of single set of chromosome plants. Single set of chromosome plants cannot develop into plantlets, cannot develop into florogenesis. Florogenesis means for flower development. If we have the means the plant have two sets of chromosome, one set of chromosome in recessive condition, one set of chromosome in the dominant condition. Which chromosomes are dominant in position, that characters are easily expressed in the plant external character. But in the pollen culture, we selected pollen grains are in only haploid condition, only single set of chromosomes. The single set of chromosomes characters are expressed in the plant external characters. So all chromosomes characters are easily identified in the pollen culture or in the androgenesis culture. See children, these are the applications of tissue culture. And one more important point from this secondary metabolites. Secondary metabolites means production of secondary metabolites by the chemicals. Some plants produce secondary chemicals as a secondary metabolites. That chemicals are easily synthesized by the root hair culture, virus-free plant productions, and somo clonal variations like crop improvement, like the genetic transformation. By these all techniques are produced secondary metabolites chemicals. Which chemicals are we need from the plants? That chemicals are easily synthesized by the tissue culture techniques. So these are the applications of tissue culture in plants.